Generating primary cellular models or organoid-like structures is key to answering many contemporary questions in biology. For example, we've been interested for quite some time in the mechanisms used by human viruses to facilitate replication in the salivary gland, as it is an important site for horizontal transmission. The development of the Salisphere-based system described in this protocol is a crucial advancement in our quest to answer these types of questions.
This technique allows us to generate primary cells from a variety of different patient samples. These cellular models are more reminiscent of the in vivo type situation, as the cells are not immortal or transformed like many cell types commonly used for in vitro experiments.
We've used this protocol to generate similar cells from mouse primary submandibular gland, and the protocol would likely be useful for other organs with similar cellular structures.
Within 2 to 4 hours after removal from the patient, on a culture plate, use autoclave sterilized dissecting scissors and surgical forceps to mince the salivary gland tissue into fine pieces at the size of 1 to 2 millimeters. Add 6 milliliters of the disc-based collagenase solution to the minced tissue. Then incubate at 37 degrees Celsius for approximately 30 minutes to 1 hour. Disrupt the tissue by pipetting with a 5 milliliter serological pipette 15 to 20 times. Incubate at 37 degrees Celsius for another 30 minutes to 1 hour. Repeat the pipetting and incubation step 2 to 3 more times or until the tissue resembles a slurry and can easily pass through the pipette opening. To coat wells with BMM, slowly pipette 500 microliters of overnight thawed BMM into each well of a 6-well plate. Slowly swirl the plate to evenly distribute the BMM over the wells. Then, incubate the coated plate.